At very small Reynolds numbers, such as those inhabited by viruses and bacteria, rates of diffusion are on par with rates of convection of a fluid. Edward Purcell calculated that the efficiency of locomotion at small Reynolds numbers was very low, and he showed that it energetically makes more sense for organisms to just sit and wait for food and respiratory gases to diffuse to them, rather than move around and try to catch food. He analogized this to a lazy cow waiting for the grass next to it to grow. At these low Reynolds numbers, movement helps the microbes find more nutrient-rich environments where they wait for the nutrients to diffuse to them. They're not actually trying to track down and catch, predate a particular nutrient. Because these animals have not undergone selection for the ability to convect either externally over their bodies or internally, there's reduced selection. I wouldn't say they have no selection for convection, but there's certainly reduced selection given how sticky the medium is, both internally and externally. That this becomes in a way a constraint that limits their body size. So they're efficient, relying strictly on diffusion, doesn't cost them anything, but they also can't get bigger. It's impossible to scale up a bacterium to the size of a tennis ball. You can see in the bottom right-hand corner of this graph I put on there, you can see how big an animal cell is relative an average animal cell of about 10 microns in diameter, how big it is relative to a bacterial cell relative to virus. So there's huge differences in the cross-sectional area and in, therefore, the surface area to volume ratio. If we're really small and have a long rod-like shape, then there's a very large surface area to volume ratio. And the requirements for these nutrients and respiratory gases, most of the machinery that require that, most of that machinery is found in the volume of the cell, whereas the diffusion is occurring over the surface. So that surface area to volume ratio becomes incredibly important. It's just not possible to scale up a bacterium without changing anything else from the normal 1 micron size up to the size of a tennis ball. Any more possible than to take a normal human being and scale them up to, the giants of Gulliver's Travels. Chordates range in size by many orders of magnitude, as we previously have pointed out. The larvae of some species of tunicates are only about 2 millimeters long, and although it's difficult to find data on their mass, assuming they have about the density of seawater, the mass has to be approximately on the order of 10 to the minus 7 kilogram, whereas a blue whale is on the order of 10 to raise to the power of the 5th kilogram. So that's many orders of magnitude. However, there is far less variation in the size of the cells that make up the body. These data are from a paper published by Hosokawa and Sekino. What these scientists did was to collect cells from various organs from a number of marine mammals and from humans. So they looked at right whale, blue whale, sei whale, a beaked whale, and a dolphin. The two graphs on the left show the distribution in cell size from two organs, liver and heart. The y-axis shows the number of cells of a specific size. The x-axis is a little bit difficult to interpret. They measured the length of a cell and the width, and then they took the average of that, and then they multiplied that average by a factor that's shown in the legend. So, for example, the average length of a liver cell for all three species, for a human and for two of the whales, was 18 microns. The width was 14 microns.
So the average of those two values is 16 microns, but then they multiplied the 16 by 3/4 to get that value 12 that you see in the where the graph peaks. The graph on the right is a little easier to interpret. So the bars, the solid bars, the black bars are the whales, and the white bars are human cells from human. And you can see that the liver cells there are averaging to be about 16 microns. So on the y-axis, that's just the average of the length by the width of each cell type. The remarkable thing about these data, I think, is that there's really only one cell that is, significantly larger in whales than in humans, and these are white fat cells. So you can see over on the far right how the white fat cells go up to the top of the graph and then actually bend over and start down again along that y-axis. This is to keep the scale so the other cells are, readily visible. And this kind of makes sense that fat cells might be somewhat different in that they have very low metabolic rates. They're a storage depot and serve as insulation, but they don't have a super high activity metabolism. You just keep putting more and more lipids in there, and I don't think there's a lot of metabolism required to maintain those lipids. Whereas muscle cells, etc., have very high metabolism. So these data suggest that there's some sort of constraint on cell size, and that's most likely related to the fact that gases diffuse over the surface, but the gases are produced within the machinery that's contained in the volume of the cell. So there's going to be a surface area to volume ratio. The bigger the cell is, the less surface area there is for diffusion to take place, CO2 to get out and oxygen to get in to the cell. So it's unknown, why these cells are so similar, but that's highly likely an explanation. So given that throughout this huge range in body sizes, the cell sizes are about the same, this means that there's got to be a pretty spectacular circulatory system to carry those gases down to the cells and spread it out throughout the body. On the left is a schematic of the cardiovascular system of either a human or a bird, and that is evidenced by the fact that the cardiac ventricle has a complete septum in it, dividing it into a right and left side. Most other animals do not have a complete septum in the ventricle. It's only birds and mammals that have completely separated the two sides of the heart. But the heart is, of course, the major pump to circulate fluids up to the lungs and then where gas exchange occurs, and then around to the body. This process occurs in a series of steps, and that's illustrated on the right-hand side. This is known as the oxygen cascade. There are 4 steps for most animals in which oxygen is moved from the environment, from the respiratory medium down to the mitochondria, where it is going to be used to produce ATP. And since these steps are in series, this is like the links of a chain. The chain is only as strong as its weakest link. In this case, the rate of flow of oxygen through this cascade is only as fast as the slowest step in this series. So the first step is a convective step. So either water is flowing over gills or air is flowing into the lungs, and there's generally a pump that's required to move that respiratory medium into or over the gas exchangers. The second step is diffusive. So once the respiratory medium comes in contact with the capillaries, then there's a diffusive step in which the gases move by diffusion from the respiratory medium into the blood. People have thought, originally it was thought there was some sort of transporter that would grab the oxygen molecules and transport them across the membrane. But there's never been transporters found, and it's pretty well accepted that it's processed for both CO2 excretion and O2 absorption, that is just passive diffusion. Once in the blood, the heart pumps this blood. There's a convective step. It's going to pump this blood out to various organs and tissues. And then there's another diffusive step. So there's convection, diffusion, convection, diffusion. This slide illustrates the airways of the male trumpet manacote. This is a bird of paradise. The spiraling structure in the chest is the trachea. It's greatly elongated in these birds due to the fact that the syrinx is located at the bifurcation of the trachea into the two primary bronchi, and the syrinx produces sound. And by the elongation of the airway downstream from the source of the sound, the pitch is altered so that the bird produces a deeper song. This may make the individual sound like a larger animal to females. This certainly arose from sexual selection, but exactly what it does beyond deepening the voice is not known.
The vascular system is also of great interest. It is very vast, and its major pump, which is the heart, has great diversity of form. To get a sense of just how vast this network is, I wanted to point out that if you place all the blood vessels from a human body, if you were to cut them out and place them end to end into one long rope, that rope would stretch all the way around the earth 2.5 times. So the bottom line to this introduction to the flow of fluids through tubes is that there are many interesting and important functions of organisms that relate to how fluids move through these enclosed vessels. To dig into the meat of these fluid dynamics, I want to first review some terms concerning pressure, because as we have already seen, the term pressure refers to several different phenomena. I will begin with a review of the concept of pressure and the concept of conservation of energy, and discuss these, apply these terms and these concepts to a stationary situation before we move on and think about flow through pipes. We're going to think about the stationary situation first. Pressure, of course, is force per area. And there's several different types of pressure, which I've listed here, which we'll talk about in the next few slides. Consider a fluid element in a glass of water that is a distant z0 from the surface. The static pressure exerted on that fluid element, which is often referred to simply as the pressure, the modifier of static is dropped, and usually represented by the letter P, is given by the atmospheric pressure P0 plus the pressure that's exerted due to the column of water above the fluid element, and that is given by the density of the water multiplied by the acceleration due to gravity multiplied by the depth. And this pressure can be measured with a mercury column or a Bourdon tube. Sometimes we don't really care about the atmospheric pressure, and that's just left off and set to 0. When that happens, that's the gauge pressure. Now let's think about the energy of this fluid particle. So the conservation of energy is a concept that applies to an ideal fluid, which water is not. An ideal fluid is a fluid that doesn't have any viscosity. But of course, water obviously does. And what it's basically saying is if you lose potential energy, that potential energy is going to be converted into kinetic energy or vice versa. So the total energy of the system is the same. With a viscous fluid, some of that energy, because of shear stresses in the water, because of viscosity, some of that energy gets consumed, it gets converted into friction and heat. But if it were an ideal fluid, then the total energy of this system is equal to the potential energy and the kinetic energy, plus the kinetic energy. But the kinetic energy in a static situation is 0. So that the total energy of this fluid element is just that potential energy. How do we compute that? You know that if we drilled a hole in that glass at the level of the fluid element, it would go squirting out with the fluid elements next to it. Right, so it's being driven out by that pressure head, that static pressure head, which is P naught rho g z1 plus rho g z1. However, because gravity is present, it is not going to just translate straight out. It's actually going to take a path in which yes, it gets squirted out, but it also flows down to the earth. And so there's a certain potential energy that that fluid element had just by the virtue of its altitude. Just how high was it above the ground? And so the total energy of the system, this potential energy, is the static pressure plus this elevational pressure, which is sometimes called the hydrostatic pressure. Let's apply some of these concepts to a fluid dynamic situation. Imagine a garden hose, water's flowing through this tube. We have the same hydrostatic related pressures that we just talked about with a glass of water, but there are some additional pressures that need to be considered. And I'm right now just talking about static pressures. If you put a hole in your garden hose that was at the top of the hose, water's going to squirt out. Doesn't matter that gravity's pulling that water down. There's an additional component to this static pressure that we need to take into account. And you could measure that if you could put a pressure sensor in the tube and it was flowing along with the same velocity as the tube, it would measure this additional pressure. The pressure is uniform. It being exerted on all the walls of the tube. It is the same in the center of the tube as it is on any circumference. And that isn't the case with the pressure head that we saw with a glass of water. That depended upon the depth of the water. This is different. This is a different pressure that must be considered. It is a static pressure, but it's an additional pressure that you have in water flowing in a confined tube like this that you don't have in water that's flowing in a river or in an open stream. A clever device that will measure this extra internal pressure is known as a piezometer. And all you have to do is to connect a side tube to where you poke the hole and seal those two tubes together. Water will gush up into the side port until the pressure that's exerted by the fluid in the side port equals the pressure pushing the fluid out of the garden hose. This pressure is sometimes known as the piezometric head. And if you wanted to compute the total static pressure at point 1, it's this piezometric head. So rho g z2 plus the pressure exerted by the column of water that's in the tube, so rho g z1. Your total static pressure then becomes rho g z2 plus rho g z1 plus the atmospheric pressure.
Imagine you're holding a garden hose somewhat back from the end so that the end droops down and is lying on the ground. The potential energy where you're holding it at point 1 is greater than the potential energy at point 4 because of this elevation. So there's ability to do work just by the fact that it's elevated up above the ground. So the total potential energy of the system at point 1, at a fluid element located at point 1, is the static pressure, which is rho naught, atmospheric pressure, plus rho gz1 plus rho gz2, plus this elevational pressure, which is sometimes also called the hydrostatic pressure, and that's given by rho g, the height at which you're holding that hose. And you'll see this written as the bottom equation shows that with a P, again, that means that's the static pressure plus the hydrostatic or the elevational pressure, rho gh. Conservation of energy says that the change in potential energy between point 0.1 and point 0.4 is going to be converted, that difference in energy is going to be converted into kinetic energy. So a fluid particle at point 0.1 is going to have a higher potential energy than it does at point 0.4, but it's going to be moving faster. It's going to have greater kinetic energy at point 0.4 than it did at point 0.1. This is true, again, for an ideal fluid, so we didn't lose any energy to viscosity, to friction and heat. And it is, you always have to compare fluid elements that are on the same streamline, so for this to be true. And so this fluid element at point 0.1 has to be compared to a fluid element that is on the same streamline. So if you wanted to know the energy of the system of where this red dot is located and how that changes with the flow, you have to compare that to the fluid element on that same streamline at a later point in time or at a different location. So this conservation of energy equation only holds true for fluid elements traveling along their respective streamlines. The dynamic pressure, the stagnation pressure, and the total pressure can be related and understood in terms of the Bernoulli theorem. We've seen the dynamic pressure before. It is the product of 1/2 density by velocity squared. And it can be measured with a device known as a pitot tube. One could stick another tube into this garden hose that has a right angle bend with the orifice facing the flow. And the momentum of the water flowing into that orifice will push the column of water in the side port even higher. This extra height is known as the dynamic pressure head. The total pressure is the pressure at point 0.4 is known as the stagnation pressure. So the water is not moving at that point, and that's why it's said to be stagnant. And it is given by the dynamic pressure plus this static pressure. So 1/2 rho, the velocity of 0.4 squared, plus the static pressure, rho g, z4 plus atmospheric. However, we know that V4 is 0, and so this simplifies to simply the static pressure at 0.4. The pressure, the total pressure at 0.1 is given by the dynamic pressure plus the static pressure. And by the Bernoulli theorem, if 0.1 and 0.4 are on the same streamline, then the energy at these 2 points of a fluid element is the same. So there is more kinetic energy at 0.1, but there's more potential energy at 0.4, but the sum is the same in these two situations. So we can set these two equations equal to each other and solve for velocity at 0.1. So the velocity at 0.4 is 0. There is no difference in the altitudinal pressure, that rho gh, since this is in this situation, we don't have any difference in height. And the atmospheric pressure subtracts off, and you get the simplified term of the stagnation pressure is equal to the static pressure at 0.1 plus the dynamic pressure at 0.1. Pitot tubes are used to measure the velocity of airplanes, and that's illustrated here on the Airbus A380. And then when they're sold commercially, the two tubes are put very close to each other. In fact, they're usually concentric cylinders. And that's illustrated on the left. So the central cylinder is open, the orifice is open and pointing into the flow. The outer cylinder is not open on the end, it's got a port on the side, and that's measuring the static pressure, and then you can find the pressure differential between those 2 points and solve for velocity. You will see the Bernoulli equation written in different forms. The form that we have been working with and thinking about is called the pressure form. So we had the static pressure plus the dynamic pressure plus this altitudinal or hydrostatic pressure equals a constant. But you can divide through, for example, by density or density and gravitational pull. And such as in the bottom, you'll get what's known as the head form. So you're just measuring the height and then able to deduce the rest of the equation. But these are obviously different constants, but you will see the Bernoulli equation written in these different forms, but it's still just the Bernoulli equation. Throughout the respiratory and circulatory system, the relative importance of dynamic pressure compared to static pressure in driving flow, whether it's airflow in the lung or water flow or blood flow in the circulatory system, depends very much on the Reynolds number and the geometry. In most of the smaller vessels for humans, the pressure that is driving flow is dominated by the static pressure. It's mainly static pressure, and one doesn't have to consider the dynamic pressure. But around the heart, this is not the case, and also in a lot of disease conditions, this is not the case. This slide illustrates a schematic of a normal human heart, and there are 4 valves in the heart, 2 between the atria and ventricle, and 2 at the outflow tracts, so the pulmonary artery and the aorta. Most of these valves have 3 leaflets. It's only the mitral valve that contains 2. There's a common congenital abnormality, however, in which the outflow tract, the aortic valve, the outflow tract between the left ventricle and the aorta, has just 2 leaflets, and I'll come back and talk about that in a second. But first I wanted to illustrate the importance of this dynamic pressure and higher Reynolds number flow at the outflow tract of the left ventricle.
Are you a professor of medicine? Then you might know that this is a heart. And 10 points if you knew that this structure is called the aorta, the main blood vessel leaving the heart, supplying the body with oxygenated blood. And the aorta features a very clever piece of evolutionary engineering that we've only recently proven, yet Leonardo da Vinci predicted correctly 500 years ago. Pressures within the aorta can be quite considerable. A normal human's blood pressure is about 120 millimeters of mercury, roughly equivalent to a swimming pool at the depth of about 5 feet. But the highest recorded blood pressure belongs to a giraffe, which reaches almost 400 millimeters of mercury. So I know what you're thinking, and it's an obvious question really. Yes, how far in the air would blood go if you cut someone's head off? Well, today we're going to find out. It seems the ethics committee had some issues with that plan, whatever, but genuinely this is a lot more interesting. See this bulge at the bottom of the aorta here, just above the aortic valve? This is called the sinus of Valsalva, named after Antonio Valsalva, who described it some 200 years after Leonardo da Vinci, the star of this video. Now, you probably shouldn't regard the turtle's intro as historical fact. Raphael was, of course, cool, but he wasn't that rude, and Donatello wasn't the engineer, techie dude. He was an artist. It was Leonardo that was the real whiz with machines. Even the great polymath Michelangelo didn't excel in such disparate fields as Leonardo, and nothing demonstrates his genius and ability to combine his interests of engineering, anatomy, science, and art as beautifully as his work on the heart. He looked at this bulge and saw it repeating in the aortas of oxen, pigs, sheep, cows, and humans, and he realized it must have a purpose. Indeed, our aforementioned giraffe has a sinus of Valsalva, as do all mammals and several other classes of animal. So what's it for? If you're friends with a doctor, try this. Ask them how the aortic valve closes. Remember, this is the one leaving the heart. They will say that blood is pumped out, slows, and then as it stops, it starts to fall back and the valve shuts. This is wrong. If that were the case, then with each heartbeat there would be some leaking of blood back into the heart, and the leaflets would crumple up and fold incorrectly. So Leonardo clearly thought about this. Years earlier he'd watched eddies formed in water that was squeezed through a narrow space, and he realized this is what was happening when the heart was pumping blood through the aortic valve. He predicted the ejected blood would possess revolving impetus. that would actively shut the valve before blood flowed backwards. He had predicted the sinus of Valsalva was shaped specifically to create vortices in order to close the aortic valve in the most efficient way possible with no leak. Leo being Leo, he built a glass model through which he pumped water. In the water were grass seeds so that he could visualize the flow dynamics, and these are his beautiful sketches showing what he saw. Incredibly, it took 450 years to prove him right. An engineer called Brian Bellhouse and his team built an updated model in 1969 which confirmed the vortices and that the aortic valve closes before the ejection of blood has finished, just as Leonardo had theorized. And just in the last few years, we're able to use 4D flow cardiac magnetic resonance imaging, which is what I spend some of my time doing. And here you can see the exact vortices Leonardo da Vinci predicted without any modern technology almost 500 years ago.
It is quite common for humans to have a congenital abnormality in which the outflow tract to the aorta is composed of 2 leaflets rather than 3. And this is these are illustrations from a paper that was where the authors were trying to understand the fluid dynamic consequences of that. So it's known that these people are at a higher risk of developing aneurysms. So the blood vessel, there's damage to the wall of the blood vessel, and the vessel breaks. So they were trying to understand why that happens, and they took pig hearts. You see, you're looking into the tricuspid valve of a pig heart. The normal heart is at the top, and what they did was they sewed 2 of the leaflets together. So they went in with suture and actually tied them together. And then they used a pump and ran blood through this aorta and looked and put pressure gauges in the aorta to understand how those stresses on the wall change. And they also followed up with they also made a computational fluid dynamics model, which you can see over on the right, of normal flow, which is shown in the first column, the normal tricuspid valve, and then the 2 different situations in the middle column and in the right-hand column where one of those valves had been sewn to another. And you can see that the dynamic pressure is much more axial. I mean, that there's much more flow axially, and there's less of a jet that's hitting the wall and causing an increased stress on that vessel wall. Consider flow of a viscous fluid. So not an ideal fluid, but one that has viscosity and that is going to resist movement and create shear stresses on the wall. In a pipe, laminar flow in a pipe, there's a pressure gradient driving this flow along the length L, and the equation for the velocity of the flow is pretty readily found. The force at steady state, so the force pushing the fluid, again, pressure is force per area, so the force pushing this fluid can be found by computing this small pressure difference between point 1 and point 2 and multiplying that by the area over which that pressure is exerted. The resistance is given by the surface area along the wall of the cylinder. Or you can think of this as concentric cylinders, because each layer, right, is moving a little bit more slowly as you move towards the wall. The velocity of the wall is 0. So each layer, as you move from the center of the cylinder out towards the wall, is moving a little bit more slowly than the layer that's closer to the center. So that shear stress can be found by this little change in velocity gradient in a little cylinder of thickness dr. That shear stress, or that resistance to flow, is related to du dr multiplied by 2 pi r L. And at steady state, these two forces, the force pushing and the force resisting, are the same. So you can set those two equations equal to each other and solve them for this velocity gradient, this little du, as a function of any place along the axis of the cylinder as a function of r. Integrating then gives this equation of r squared multiplied by the pressure gradient delta P divided by the product of 4L viscosity plus some constant. But we know some boundary conditions, so we can solve for that constant. The flow at the wall is 0. So when the radius is equal to the total radius of the vessel, A, then we know that the flow is 0. So using that, you can solve for this constant C, and then you get this equation that describes the velocity profile in the pipe. And this checks out in the sense that when the radius is equal to A, then that quantity, the value in the brackets is going to be 0. When r equals A, then you get 0, and that says there's no flow at the wall. So that checks out. And also the smaller, when r is 0, that's going to give you your maximum velocity. So that's right in the center of the tube.
The shape of this velocity profile is a parabola, as shown here. So the flow in the center of the tube is fastest, and at all the edges it drops down to be 0. One can use this velocity profile now to find the volumetric flow, so not the velocity of the fluid moving through the tube, but the volume. Once the velocity profile is known, then it is a pretty straightforward thing to calculate the total volumetric flow. And the approach would be, since it's symmetrical, you can consider a concentric cylinder at some location r, away from with a radius of r. The volumetric flow through that little cylinder is proportional to the velocity profile, the velocity at that point r, multiplied by the cross-sectional area of that little cylinder. So that's 2 pi r dr. So plug in the equation for the velocity profile at that point, and that will give you the little volumetric flow going through that cylinder. And then to find the total flow through the cylinder, you integrate from 0 to r over that 2 pi r dr u sub r. And you get this equation that is highlighted in blue, where the total flow is equivalent to the product of pi and the pressure differential over the tube and the radius to the 4th power, divided by the product of 8L, or the length, and the viscosity. And this equation makes sense if you think about keeping the pressure gradient the same. If you increase the viscosity, flow is going to go down. If you increase the length and have the same pressure gradient, flow is going to go down. So the surprising part of this equation, and one of the important things to notice, is that radius to the 4th power term. That means that little tubes have a disproportionately less flow going through them. And that's because the viscosity is arising from the walls of the tube, the walls of the vessel. And so there's more surface area to volume ratio, so there's going to be more drag. You're going to have less flow going through that for a given pressure gradient. This relationship is known as the Poiseuille or the Hagen-Poiseuille equation, and it is a very important relationship. Turbulent flow has a very different velocity profile. It is not this parabolic shape. It is flattened. You have completely different shape to that velocity. So the velocity near the wall is much closer to being the same as compared to the velocity in the center of the tube. There's another important difference. The resistance to flow is much greater with turbulence. The Poisson equation gave us a relationship between the pressure gradient and the resistance in the pipe, the viscous resistance and the flow. And that relationship for laminar flow is linear, as we just saw. Increase the pressure gradient, double that, you're going to double the flow. And double the resistance without increasing the pressure gradient, the flow is going to be cut in half. So that's a linear relationship as shown here. So the pressure gradient for the laminar flow is on the left-hand side, and any increase in the pressure gradient produces a, proportional increase in a linear increase in the rate of flow. But look what happens for the turbulent flow. So at low flow rates, you get a disproportionate increase in the rate of flow for a small increase in pressure gradient. Right? It's not a line there. And it is, and then when you start getting to higher and higher flow rates, now you have to have a disproportionately big pressure gradient to increase the flow just a little bit. So that's a nonlinear function. I've written some values for wind flow transitions from laminar to transitional to finally turbulent. And these are very rough estimations because other things matter. As we know, surface structures can matter, whether it's rough, if you have a rough surface that can cause turbulence at a lower Reynolds number, and other things about the geometry, like are there bifurcations and things can affect this, whether or not turbulence sets in. Also, this is different for pulsatile flow. We're talking about steady state flow here in all of these, in this lecture so far.
I want to make one more important point about resistance to laminar flow in a vessel. I already pointed out that this resistance varies by the radius raised to the fourth power. This is illustrated in this figure. So if the radius is 1 and a pressure head driving flow is 100 millimeters, in this example, the flow through the tube is 1 mil per minute. If the radius is doubled to 2, that increases the flow 16-fold. And if the radius is doubled again to 4, the flow increases another 16-fold to 256 mils. So in a 4-fold increase between vessel 1, the top vessel, and the bottom vessel, you had 256-fold increase in flow. The difference between the radii of a capillary in a human and the aorta is 3,000-fold. So a capillary, or the aorta, is 3,000-fold greater than a capillary. So you can imagine how much difference there is in resistance between blood flow through one human capillary and the aorta. However, if you look at the pressure drop, and we know that the pressure drop is given by the flow divided by the resistance, it's a function of the resistance. If you look at the pressure drop across the cardiopulmonary circuit, the entire circuit, what we see is that there is not a big pressure drop difference between the capillaries and the aorta. Not nearly what you would expect because of this 3,000-fold difference in radii. And the reason is because this is across the entire capillary beds, and there's there's a lot of capillaries. And when you add up resistance that's in series, the resistance simply adds. However, if you're adding the figure the total resistance across a bed where the vessels are arranged in parallel, those add as shown in this equation. 1 over the total resistance of the entire bed is equal to 1 over each individual resistances. And so that explains why we don't have a huge pressure drop across the capillary bed, not nearly as big as you might think from this R4 factor. These are data for the airway dimension velocities. So this would be the average velocity in the airway, and the Reynolds numbers for the parts of the human respiratory tree. The trachea is about 2 centimeters in diameter, and the Reynolds number at rest for quiet breathing, so this is a flow of about 0.5 times 10 to the minus 3 meter cubed per second. The Reynolds number is around, a little over 2,000. So you're getting into transitional flow in the larger airways during quiet breathing, and you definitely have turbulent flow with vigorous breathing throughout a number of the branches of the bronchial tree. At rest in humans, the Reynolds number is low enough in the vasculature that there shouldn't be turbulence. This table shows the radius of the vessels and the Reynolds number for resting conditions, so a heart rate of 60 beats per minute. You can see that 1500, it's getting close, but it really shouldn't be a lot of turbulence. Stenosis or other problems in a vessel can also lead to abnormal flows, which can be problematic, leading to rupture or clots, or just the vessel ending up being damaged enough that it's, it closes off. I like this saying by a doctor. Here's a little saying. He says, "Streamlined flow is silent. Remember that, my boys. But when the flow is turbulent, there's bound to be a noise. So when your stethoscope picks up a bruit, murmur, sigh, remember that it's turbulence and you must figure why." Blood is not a Newtonian fluid. Recall that a Newtonian fluid is one in which there is a linear relationship between the shear stress and the velocity gradient. If you look in the left-hand corner, you'll recall that water or blood is moving more slowly near the wall, and there are layers in which those layers are increasing in their velocity. This would be the U is represents velocity here. So as you move to greater values on the y-axis, you come closer and closer to a free stream velocity, or in the case of a vessel, you're going to come closer to the center, the velocity in the center of the stream. And the shear stress, the stress that's exerted on the wall, is given by this equation of viscosity multiplied by that rate of shear deformation. Now, so that's a normal Newtonian fluid, but blood doesn't behave this way. So when the blood is in a very large vessel like the aorta, the deformation of the red blood cells works to reduce the viscosity of the blood. So that this saves in terms of work of the heart. It's easier for the heart to pump blood because it appears less viscous. It is less viscous in a large diameter vessel like the aorta. In vessels that have a diameter between 30 and 300 microns, there is an effect in which the plasma, the blood cells, are mainly traveling in the center of or closer to the center of the vessel, and the plasma is what's coating the edge, and this plasma is not moving as fast. Right? If this is laminar flow, we know you have a velocity profile, so the fluid in the center of the tube is moving faster. So you have this situation, an interesting situation, where the red blood cells are moving faster than the plasma. So they're circulating through or passing through the vessel at a higher rate. And then below 10 microns, then the red blood cells start passing through in single file. And if it's very much below 10 microns, then they're
the blood vessel is actually smaller than the red blood cells, and so the red blood cells have to deform in order to squeeze through those capillaries and to pass through those vessels. When you get down to these smaller capillary size ranges, the blood gets more viscous. So we definitely have, it's stickier in going through. So we definitely have non-Newtonian fluid when we are talking about blood. But part of this increase in viscosity in these smaller vessels, it probably has to do with proteins that are coating the endothelial cells that are lining the vessels, that are interacting with proteins on the red blood cell. When fluid flows in a curved tube, it is subjected to centrifugal forces. These combine with the viscous forces established by the solid boundary of the walls to create a second set of flow patterns. These patterns can be very important in terms of particle deposition and also in terms of mixing. I'd like to show you a little clip of one of these secondary flow patterns that's set up in an open tank where the fluid is moving in a curved path. I think it's open, so it's not exactly the same as flow in an enclosed tube, but I think it gives you a pretty good idea of how these secondary patterns are set up.
The flow in this tank is a combination of a vortex and a sink. Water enters tangentially here and flows out at the center. The question is, what path does the water take from the inlet to the outlet? Here I have a long-nosed hypodermic with dyed water in it. With this I can trace the flow and see where it goes. Let's try a little at the surface and see what's happening. At this rate, it'll be a long time reaching the drain. Maybe it never will. So much for surface flow. Let's start over. This time, we'll try injecting at about half depth. Again, the pattern seems to be one of nearly circular flow. The water going out of the drain must come from the bottom. A small spur holds the tip of the needle about 1/8 of an inch off the bottom. We can see that the dye goes rapidly toward the center, and this seems to be the answer to our question. We've examined the flow here, which behaves differently at different depths. At the surface, the flow is nearly circular, and this situation persists well down into the flow. This nearly circular pattern could be called the primary flow. Near the bottom, however, the situation is different. Here the flow sweeps inward in a steep spiral and down the drain. This departure from the general situation is called secondary flow. Let's see if we can get a little more specific idea of what we mean by primary flow and secondary flow. In order to understand a flow, it's often useful to think of it in two steps. The first step is to approximate the flow by a description which fits the major part of the flow with reasonable accuracy. This first approximation we call primary flow. The second step is a refinement of the first approximation. It is a recognition of the difference between the actual flow and the first approximation. It is this difference that we call secondary flow. I can stir my tea so as to cause a circular motion. Inertia keeps this primary flow going. The tea leaves are a little denser than the liquid and settle slowly toward the bottom. Since they are denser, we might expect centrifugal force to throw them outward and to find them around the perimeter of the bottom of the cup. But look, they collect at the center. The cup has no drain, but still we find evidence of a flow toward the center near the bottom. We've made an experimental model of this flow in order to examine it more closely. This square box has a circular container inside. It has been rotating around its axis for a couple of hours, bringing the liquid inside to a state of uniform rotation. I can stop the rotation, but the liquid, due to its inertia, will continue to rotate at uniform velocity, except near the wall and the bottom, where a boundary layer will build up. As a first approximation, we will assume that all particles travel in circles around the center of the cylinder, making allowance for the fact that the boundary layer moves at a slower rate than the rest of the flow. We will call this first approximation the primary flow. In order to observe this flow better, we have stretched a very fine wire vertically to the bottom of the tank. Hydrogen bubbles electrolyzed from this wire are stripped off by the flow. By pulsing the current, we can create a series of bubble lines. These lines show up very much better if we dim the room lights. From above, it appears that much of the flow does indeed follow a circular path. Near the bottom, however, a portion of the flow appears to go inward. We can now look into the tank at a tangent to the flow past the wire. And incidentally, we can see that the reason for the square box is to eliminate optical distortion. In this tangential view, we see that the simple circular flow occupies the greater part of the depth. The inward flow extends only a short distance from the bottom. This inward flow is a departure from the assumed primary flow and is what we call the secondary flow. And now let's look inward in a radial direction, that is across the streamlines. We see the circular flow as before and the effect of the boundary layer near the bottom. But notice that this boundary layer appears to affect about the same portion of the total flow as did the inward curving secondary flow. If we look at the radial and tangential views side by side and in the same scale, it becomes clear that this secondary flow occurs within the boundary layer. The inward movement in the boundary layer seems to be directly associated with the reduced tangential velocity in the boundary layer. Our simplified model has given us some insight into this particular secondary flow. Is it the reduced tangential velocity in the boundary layer which results in the spirally inward flow? Let us consider a fluid particle moving on a circular streamline. The pressure force on the outside must be larger than the pressure force on the inside in order to result in a net pressure force inward. The pressure gradient, you will recall, is described by Euler's equation, normal to the streamline. Suppose that a particle in this pressure gradient has higher than normal velocity. It will then have to follow a larger radius and be carried outward. If it has a smaller velocity, it will have to follow a smaller radius and be carried inward. In our experiment, the pressure gradient is established by the main circular flow and impressed on the boundary layer below. The slower moving particles in the boundary layer find themselves in just this situation. Same pressure gradient, but lower velocity, and therefore are swept inward. Tea leaves in the bottom of the teacup are carried to the center. Dye in the bottom of the sink vortex sweeps inward. A predictable secondary flow explains each phenomenon. Suppose we invert the sink vortex by taking the water from the top instead of the bottom. I have closed a drain at the bottom and arranged this siphon to withdraw water from just below the surface. And now I will put some dye near the bottom in the boundary layer as before. You ever see a tornado? Here's another piece of apparatus for investigation.
This illustrates these secondary flows that are set up in a tube, an enclosed tube, a blood vessel or tube in the airway, for example. So again, the fluid is pulled out towards the outer edge of the bend, and when it hits the wall, the flow splits. Some of that flow starts moving up around the top of the tube, and some of it starts moving down around the tube, so you get these secondary eddies set up. This is the same thing, just showing you from a little different angle. So the fluid, as it goes around the bend, wants to be pulled out towards that outer edge. When it hits the edge, some of it moves up and starts circulating around the top of the tube, and some of it moves down and starts circulating around the tube. These secondary motions are important in terms of the shear stresses are different than they would be in a straight tube. They also mix the contents of the center of this tube with the edges, and so if there's diffusion, for example, of respiratory gases or heat, or this becomes important as a source of mixing. It's also important in terms of particle deposition, in terms of where maybe aerosols will actually come in contact with the edge of the respiratory system, for example. Another factor that sometimes needs to be taken into account is that blood flow can be pulsatile, and airflow in the lung is pulsatile. It's not a constant pressure gradient driving that flow. Now the degree of pulsatility depends very much on the vessel. So, for example, I have a slide of an image of the aortic pressure. So this is starting with a contraction of the ventricle. You have isovolumic contraction, and then the valve opens and there's a period of ejection, and then the valve closes, and then the heart is relaxing. So this is systole and diastole in this pressure phase of the aorta. And the coronary arteries, which come off of the aorta, very close to the outflow tracts. This is a blood flow. This shows blood flow in the left coronary artery. And you can see that during systole, there's a very little flow in the coronary arteries because the heart is contracting and increasing the pressure, squeezing the vessels in the heart, squeezing all that vasculature in the heart. So there's a little blood flow during systole, but quite a large pulse during diastole. In other vessels, this would be the opposite. You're going to have a large pulse in the aorta during systole, and a smaller pulse during diastole. The relationship between the vessel size and the pressure profile that's driving the flow and the velocity of the fluid in the flow is given by what's known as the Wormsley number. So this is a dimensionless number that is very much like the Reynolds number, and it's represented by the letter alpha, and it states that the alpha to raise to the power 2 is equivalent to the density of the fluid, the frequency of the pressure profile, the pump, the frequency of that, so that would be heartbeat in the cardiovascular system, multiplied by the speed of the fluid in the vessel, divided by the viscosity, divided by, again, the speed, and a characteristic length, which is in blood vessels, it's the diameter of the vessel. And this can be reduced to a simpler form, which is given here, the characteristic length of the vessel, this frequency of the pump divided by the kinematic viscosity to the 1/2. So the bottom line here is that this is very much like the Reynolds number. If you have a large Wormsley number, then inertial forces are dominating, and if you have a small Wormsley number, viscous forces are dominating and driving your flow. And I've given some examples of Wormsley number for different sized vessels. So anything that's smaller than 1, that's around the value of 1, the pulsation is sufficiently low that you have this Poiseuille's profile in this parabolic velocity profile, and a pretty constant flow. So there's not a lot of pulsations. I've seen flow through, say, the pulmonaries, capillaries, and I basically see no pulsation in the flow in vessels when you get down to these very small Wormsley numbers. But obviously there is pulsation in both pressure and flow in other vessels. So this is telling us in these smaller vessels, viscosity is dominating. You don't have as much inertia that is driving flow, and the phase between when pressure is showing up in the vessel and when flow is occurring in the vessel is pretty well linked. But when the Wormsley number is higher, there can be a decoupling of the pressure profile and the flow, because there's an inertia to the flow, right? So it's going to, even after the pressure has dropped in a vessel like the aorta, the flow is going to continue for a while because it still has momentum. It'll take a while for those viscous forces to slow it down and stop it. And so there isn't necessarily an exact correlation in time with when the vessel is experiencing its peak pressure, pulse, and the peak flow. So this Wormsley number can be quite important under some circumstances, and it becomes, I think, even in these smaller vessels, if there's some sort of occlusion, stenosis, then you can get these very high pulsatile flows. And in fact, I've seen papers where they've modeled vortex rings forming from these pulsations coming through very small when the vessel is occluded enough. So you remember that the very first movie I showed at the beginning of this lecture with that gun that was shooting off the vortex rings. So you can get these vortex rings that are shooting down the blood vessels if the stenosis is organized in the right way. I'd like to turn now to a short discussion of diffusion. We already mentioned that there is a diffusive step in the oxygen cascade from after a respiratory medium is convected up to the gas exchange tissues. Then the next step is diffusion from either water or air into the blood across a series of membranes. And then the heart pumps the red blood cells out to the tissues. And again, there's another diffusive step for those gases to exchange between the tissues and the blood. So I'd just like to talk a little bit about the history of diffusion, who played an important role in thinking about it, and notable role in thinking about it, and talk a little bit about what determines its maximum rate. What are the important parameters, and anatomical parameters that are important to determining this diffusive step? And again, this is important in that these steps occur in series. So the slowest step will set the flow of respiratory gases through the entire cascade. So diffusive steps need to keep up with the convective steps. It is incredible to me that the idea that there are very small invisible particles that are bouncing around and hitting each other and moving in this invisible realm actually goes back to the Greeks hundreds of years before Christ. And one of the earliest philosophers, scientists who thought about this was Aristotle. And he claimed that the atoms were indestructible and immutable and moved through a void, bouncing off each other and sometimes becoming hooked together to form clusters. And then Lucretius, in his book on the nature of things, wrote, "Observe what happens when some beams are admitted into a building and shed light on its shadowy places. You will see a multitude of tiny particles mingling in a multitude of ways. Their dancing is an actual indication of underlying movements of matter that are hidden from our sight. It originates with the atoms which move of themselves, spontaneously. Then those small compound bodies that are least removed from the impetus of the atoms are set in motion by the impact of their invisible blows, and in turn cannon against slightly larger bodies. So the movement mounts up from the atoms and gradually emerges to the level of our senses, so that those bodies are in motion that we see in sunbeams, moved by blows that remain invisible."
Brownian motion. Here you can see tiny plastic beads in water seen through a microscope. The beads are only 1/1000th of a millimetre across. In 1827, scientist Robert Brown was studying pollen grains in water. He noticed that the grains of pollen were constantly moving. They jittered and moved about randomly. He suggested that the pollen grains were moving because they were alive. He repeated the experiment with soot grains which he knew were not alive, but he saw that they moved in the same way. He could not explain what he had seen. These are soot particles floating in air. In 1905, Albert Einstein proved that the movement was caused by tiny, invisible, fast-moving particles hitting the pollen grains.
The next person I want to talk about and his contributions to the theories of Brownian motion and diffusion and what are known as random walks is Einstein. In 1905, Einstein published a paper titled "Investigations on the Theory of the Movement the Brownian movement, in which he worked out mathematically how to determine the speed at which a molecule will move from point A to point B with this so-called Brownian walk. So he based this on thermodynamic principles, and this is the bottom line of that equation. That is, the root mean square of the distance is equal to twice diffusion coefficient multiplied by the time. So this expresses the displacement in terms of the time elapsed and a value that you can look up in tables known as the diffusivity. And that has to do with the diameter of the particle, primarily, of the atom or of the molecule. But this important part here is that it's related to the square root. The time is related to the square root of the distance. So it's not a linear relationship between time and distance as we normally think of velocity. This shows a little bit of the kinetic theory of energy. 3/2 kT is the energy of a particular molecule, and this gives values, and just in case you're interested in a little more of the background of that paper. But basically, at room temperature, what this is saying, at room temperature, molecules have a heck of a lot of energy. So if you plug in values for the mass of a molecule of oxygen and room temperature, 20 degrees Celsius, then what you find is a molecule is traveling on average at 473 meters per second. So if it were unimpeded, if there was not any other molecules or atoms to run into, to bounce into, it could do a 100-meter dash in little more than 1/5 of a second. So they're moving very fast, but they are not moving unimpeded. And so because they're bumping into all these other molecules or atoms, the actual time it takes for them to go very short distances is quite long. Rates of diffusion of not an individual molecule, so we're not talking about the random walk and Brownian motion anymore, but we're talking about rates of diffusion of a bunch of molecules on one side of a barrier compared to another. Those are dependent upon the partial pressure difference. This is not the same pressure that we've been talking about when we're thinking about convective flows, right? Because that was all the pressure exerted by all the atoms or molecules. But this is just that particular type. Let's say it's oxygen. The pressure differential between the point at P1, maybe that's the air in the lung, and the point at P2, let's say that's the blood. That driving gradient then is important, but also how far, the distance in between the point P1 and P2. And that's inverse, the rate of gas flow is inverse related, or the rate of diffusion is inverse related to that. So the thicker, because of the Brownian walk issue, the thicker the membrane, or the thicker the barrier, the slower gases or molecules will diffuse from P1 to P2. It also is directly proportional to the cross-sectional area of the membrane. The more surface area there is to diffuse across, the higher the rate of diffusion. And then again to this coefficient of diffusion, and that has to do with, like I mentioned previously, the size of the molecule, but also some other factors like they're lumped in a gas solubility. So does the molecule have a big dipole moment? If it does, it's going to really be happy to dissolve in water. If it doesn't, and oxygen doesn't, then it's doesn't want to fit in with the water molecules. So there are other properties besides just the size of the molecule that go into this diffusion coefficient. But the important point, take-home point here is that rates of diffusion of gases across the respiratory surface or across the surfaces from blood to into the mitochondria, right? Those rates depend on, first, in a linear way, the surface area over which the gases can diffuse, the partial pressure difference between point 1 and point 2, and the inversely related to the thickness of the barrier. To conclude, I would like to show you some data out of a paper that shows this, how this combination of factors can be really important in terms of evolution, in terms of form and function. So these data are from a paper by Stephen Perry and published in 1993. And on one axis, this is a log plot. So we have to be careful. It's not linear, right? It's logarithmic. On one axis is the respiratory surface area. So how big, how many capillaries are actually in the lung? Basically, is what that's driving at. And the more the surface area there is for gas exchange, the farther up towards the apex of this graph, you would find your animal, the study. And then on the other axis, there's the barrier thickness. And again, it's a log plot. So there's a little movement on that graph means a big difference in thickness. And then the data for many different types of vertebrates are shown in the graph itself. And the thing that I find quite remarkable about this is that, we tend to think of, well, a lot of people think of sort of birds as having this really exceptional respiratory system. And in fact, they have a high capacity for oxygen to diffuse over those respiratory surfaces, but it's not actually that much higher compared to mammals if you really sort of look at the broad distribution of the data. So let's look at non-avian reptiles to begin with. They're for the most part lumped down here, and there are some outliers. So one is illustrated by this croc. These are the crocodiles, right? They're kind of an outlier. But we get tegus and monitor lizards and other things, and they sort of clump right here. So if we wanted to compare the performance of the reptile lung in terms of capacity to take up oxygen to that of a mammal, what you see is that most of the mammals, there's some outliers here too, but most of the mammals have about an order of magnitude more surface area in the lung. That is a major difference between reptiles and lungs. So they're going to have a higher rate of diffusion, largely because they have a lot more capillaries. They have a lot more surface area in the lung. But if you look at the thickness, this barrier thickness, it's pretty similar. The overlap is pretty similar. Maybe mammals are a tiny bit thinner, but they're just clearly not a huge difference. There's overlap between the thickness of the barrier with mammals and the thickness of the barrier of some reptiles. The difference, a striking difference between non-avian reptiles and birds are reptiles, right, is that they not only have more surface area in the lung, but they also have really thin this blood gas barrier, right? But the rates of diffusion are a combination of these things. So this, if you take the surface area and you divide it by the thickness, you get what is known as the anatomical diffusion factor. And this is plotted on this vertical scale.
And what you see is if you just, and this is the bottom line for gas exchange, because it isn't just one or the other. It isn't just thickness or surface area. It's the combination of those that is going to really determine the rates of diffusion. And so if you look at the bottom line, the rates of diffusion on this anatomical diffusion factor scale, what you see is that there isn't really very much difference between mammals and birds. They've accomplished the same end goal, but they've done it in different ways. And you also see that the outliers to the majority of the groups are animals that have distinct modes of locomotion. So bats clearly are superior to any other mammal. And penguins down here are clearly inferior to most other birds. And galliforms are down here, right? And so what you have is that there's another factor playing into the design of the respiratory system that is based on mode of locomotion. So animals that are aquatic or semi-aquatic, locomotion is cheap because they're buoyed up by the water. And so it's just, it's not as hard to locomote as it is with a hummingbird. A hummingbird's going to be up here. To support your body weight in air and do this, the type of locomotion that the hummingbirds do means that you just need to produce more ATP and more power to your locomotion than if you're floating along in water. And so if you were to look at mammals, the outlying mammals would be aquatic mammals. They don't have the same demands placed on their respiratory system, whales and seals and dolphins and porpoises. So they're going to be low aquatic birds like penguins, or birds that don't fly very much like galliforms, that rely more on running. They're down here with running mammals. So there's a mode of locomotion factor that is strongly influencing where selection has driven this anatomical diffusion factor.